Inside every cell in your body, there's a tiny clock ticking away, or at least that's what scientists thought for decades. But in 2025, everything changed. For years, we believed we had a master clock in our cells, ticking away and causing us to grow old. And that idea was comforting. If aging was controlled by a clock, maybe we could rewind it. Scientists even found what they called epigenetic clocks, patterns of chemical tags on your DNA that could pinpoint your age almost exactly, like rings in a tree trunk. These clocks were more than just timers. We started to suspect they might be the cause of aging. Turn back the clock, and perhaps you could turn back aging itself. But a groundbreaking study in 2025 by Koch and colleagues just flipped this whole story on its head. It turns out those biological clocks aren't the puppet masters we hoped they were. They're more like record keepers. In other words, they don't control aging at all. They track something deeper going on. The research published in Nature Aging suggests that the clock's ticks are set in motion by something far more random and insidious: DNA mutations, tiny random damage to our genetic code that accumulates over time. To understand how shocking this is, let's rewind a bit. There have long been two big theories about why we age, and they both involve DNA. One theory said aging is caused by random mutations, little errors and breaks in our DNA that build up over time. The other theory argued aging is driven by epigenetic changes, those chemical tags on DNA that turn genes on and off, gradually drifting into the wrong pattern as we get older. Think of mutations as damage to the hardware of a computer, while epigenetic changes are like software glitches. For years, scientists debated, is it the hardware breaking down or the software messing up that makes us age? Epigenetic clocks, the software markers, then became a hot topic, and for good reason. They could predict a cell's biological age very accurately. Companies sprang up aiming to reset these clocks. Biohackers and researchers tried all sorts of tricks to make their cells younger, according to those DNA patterns. It was as if we'd found the body's ageometer, and everyone wanted to dial it back to zero. Major labs and startups bet big on therapies to wipe those epigenetic tags clean and rejuvenate cells. But here's the twist. Koch and his team discovered that those epigenetic changes, the clock ticks, are not the start of the story, but the end of it. They're responses, echoes of something more fundamental. By analyzing genomic data from over 9,300 people, the researchers found a stunning link. Random DNA mutations and the epigenetic clock were moving in lockstep. A single mutation, a tiny error in the DNA, could cause a cascade of epigenetic changes across the genome. One DNA blip might flip dozens of those gene switches, not just at the spot of the damage, but stretching across thousands of genetic letters around it. It's like a pebble tossed into a pond, sending ripples far and wide. In fact, these DNA damage patterns were so telling that the team could predict a person's age almost as accurately by looking at their mutations as by reading their epigenetic marks. The clock and the mutations told the same story. Therefore, the epigenetic clock isn't what we thought it was. It's not the driver, it's the odometer. It's tracking how much wear and tear your DNA has accumulated, not causing it. The new study demonstrated for the first time that the predictable changes in those DNA tags are tied directly to random genetic mutations. Just like the rings of a tree don't cause the tree to age, but record years of drought and rain, these epigenetic marks record the history of damage in your cells. Resetting them is like repainting a scratched-up car. The car might look shiny again, but the engine, your DNA, still has all those miles on it. This revelation has huge implications, and it's a wake-up call for anti-aging science. If random DNA mutations are the true engine of aging, then many of our current strategies have been aimed at the wrong target. Remember those efforts to reprogram cells to a younger state by wiping away epigenetic marks? They might have been treating the symptom, not the cause. It's as if we had a leaking roof and we kept mopping the floor. The floor looks dry for a while, but the water keeps dripping in. To fix it, you must repair the roof itself. But fixing the roof, our DNA, is a much tougher job. Ideker and colleagues caution that if we focus only on resetting the epigenetic clock without repairing DNA, we're just masking the problem. The real culprit is still at large. So what does this change about our quest to slow or reverse aging? Some approaches will need a serious overhaul. If aging is propelled by countless random mutations, then simply tweaking gene expression won't be enough. We'll have to prevent or repair the mutations themselves. That sounds hard, and it is. Co-author Stephen Cummings didn't sugarcoat it. If mutations are the fundamental driver of aging, it's going to be a lot harder to reverse aging than we hoped. And yet, this clarity also gives us a more effective path forward. It tells scientists where to aim our efforts, at the DNA damage. Instead of trying to turn back an illusory clock, we can focus on fortifying the genome, improving our cells' ability to repair DNA, and clearing out or rescuing cells that accumulate too much damage. At first, this might feel like a blow to our dream of easy anti-aging fixes, but in another sense, it's a relief. We've been saved from barking up the wrong tree. Knowing that epigenetic change is mostly a side effect means we won't waste as much time on flashy clock reset tricks that ultimately don't extend lifespan. Instead, we can put more energy into strategies that address the real problem. It's like a detective finally identifying the true culprit after years spent on a false lead. Frustrating, yes, but now the case can really move forward. And moving forward may involve completely new kinds of anti-aging therapies that we hadn't fully prioritized before. Imagine treatments that act like microscopic maintenance crews for our cells, constantly repairing DNA breaks and correcting errors as they arise. This sounds like science fiction now, but so did sequencing the human genome or editing genes a few decades ago. Already, some scientists are looking into ways to boost the DNA repair systems that our cells have, or to remove cells that are too damaged to function, so your body can replace them with healthier cells. We might one day use stem cells or gene therapy to replace or fix the most damaged cells in our bodies. Instead of just retuning the epigenetic software, we'd be refurbishing the hardware itself. Rather than turning back a clock, we'd be truly rejuvenating the body's infrastructure. Now, let's zoom out for a moment. In a way, this discovery reframes aging as a battle with entropy, the universal tendency towards disorder happening inside our DNA. That might sound intimidating, like we're up against a fundamental law of physics, and it is a daunting challenge. But humans are not new to battling entropy. We do it every day when we maintain machines, heal illnesses, or even just clean our rooms. We can't stop entropy, but we can fight its effects. Perhaps conquering aging means getting extremely clever at repairing and maintaining our molecular machinery. Maybe the first person to live to 150 will be someone who benefits from therapies that keep patching up DNA damage almost as fast as it happens. Everything you know about aging was wrong, and that's a good thing. Now we can finally get it right.